Good day everyone, I'm RIVERA and for today I will be discussing C3, C4, and CAM plants. So before we delve into the differences between the three photosynthetic systems, let us first recall what photorespiration is. Photorespiration is a metabolic process in plants that occur within the Calvin cycle, wherein ribulose 1,5-bisphosphate carboxylase or oxygenase, which from this point forward will be referred to as rubisco, that is the key enzyme for carbon fixation, adds oxygen to ribulose 1,5-bisphosphate or RUBP instead of carbon dioxide. This is wasteful because essentially this just undoes the work of photosynthesis, which is building organic molecules, specifically sugar. Unlike photosynthesis, photorespiration takes up oxygen and liberates carbon dioxide. This usually occurs when there is scant carbon dioxide available for rubisco to act on. To counter this wasteful metabolic pathway, plants have developed adaptations. Most plants undergo C3 metabolic pathway, which is basically the standard mechanism for carbon fixation. This is named as such because of the 3-carbon compound 3-phosphoglyceric acid or 3-PGA that it produces. This occurs in the plant's mesophyll cells. Plants that use this type of pathway include rice, wheat, soybeans, and most trees. Next are C4 plants. In these plants, reactions occur separately. So light-dependent reactions occur in the mesophyll cells of plants, while Calvin cycle occurs in the bundle sheath cells. As the name suggests, a simple 4-carbon organic acid called oxaloacetate is formed from the fixation of atmospheric oxygen by a non-rubisco enzyme, phosphoenolpyruvate carboxylase or PEP carboxylase. This enzyme has no tendency to act on oxygen, so the chances of photorespiration occurring is minimized. Still, inside the mesophyll cell, oxaloacetate is converted into malate, which can then be transported into the bundle sheath cells, where it will be broken down to release a molecule of carbon dioxide. Then, from here onwards, carbon dioxide will be fixated by rubisco and made into sugars via the Calvin cycle, as in the C3 pathway. So plants that utilize this pathway include sugarcane and corn, and these plants are commonly found in habitats or regions with hot conditions. Lastly, the CAM or crassulacean acid metabolic pathway, whose name comes from the family of plants from which it was first observed, the Crassulaceae. So unlike the C4 pathway wherein the location where the reactions occur are separate, in the CAM pathway, the reaction occurs in separate times. So during the night, CAM plants have an open stomata to allow carbon dioxide to diffuse into the leaves. So the CO2 is then fixated into oxaloacetate by PEP carboxylase as in the C4 pathway, and then converted into malate or other types of organic acid. This will then be stored in the plant's vacuoles until the next day. And then during the day, CAM plants have a closed stomata but still undergoes photosynthesis. So carbon dioxide is released from the stored malate and then enters the Calvin cycle to produce sugars. This pathway is used by plants that dominate very hot and dry areas. These include cactus, orchids, and agave. So to sum this topic up, plants that live in areas with limited carbon dioxide supply use modified photosynthetic pathways to avoid wasteful process of photorespiration. C3 plants undergo carbon fixation normally without any features to combat respiration. C4 plants separate the initial carbon fixation in the Calvin cycle to occur in the mesophyll cells and the bundle sheath cells of plants respectively to avoid photorespiration. And lastly, CAM plants separate the reactions between different times of the day to minimize photorespiration. That is all for my topic. Thank you so much for listening, and I hope that you have learned a lot. Thank you.